Welcome to Module 1 of Environmental Toxicology. In this module, we will explore the historical background, tasks, and scope of toxicology, followed by the cellular and molecular foundations of toxicology, the concepts of toxicokinetics and toxicodynamics, general toxicology and risk assessment, exposure assessment and biomonitoring, and finally reproductive and developmental toxicology. This foundation will prepare us for the applied aspects covered in Module 2. 1.1 Introduction to Environmental Toxicology. Toxicology is the science of poisons, studying how natural and man-made substances affect living organisms. The word comes from the Greek toxikon, meaning arrow poison. Throughout history, humans have known about the toxic effects of natural substances such as plant alkaloids or heavy metals. However, the modern discipline developed in the 20th century, especially after industrialization increased the release of chemicals into the environment. Tasks and scope. Environmental toxicology focuses on how contaminants in air, water, soil, food, and consumer products affect human health and ecosystems. The scope includes identifying hazardous substances, studying their mechanisms of action, assessing risk to humans and wildlife, developing strategies for prevention and control. Ethical principles. A central principle in toxicology is ethical responsibility. Scientists must balance the need for chemical innovation with protection of life and the environment. Ethical toxicology requires honesty, avoidance of harm, use of alternatives to animal testing when possible, and application of the precautionary principle when data are uncertain. 1.2 Cellular and molecular toxicology. Toxicity begins at the molecular level. When a toxicant enters the body, it may interact with proteins, membranes, DNA, or enzymes. Key molecular mechanisms include binding to receptors. Certain toxins mimic or block normal signaling molecules. Oxidative stress. Many pollutants generate reactive oxygen species that damage cellular components. DNA damage and mutations. Some chemicals cause genetic changes, potentially leading to cancer. Interference with cell signaling. Toxicants may disrupt communication pathways within or between cells. On the cellular scale, toxic effects include necrosis, apoptosis, altered metabolism, or disruption of cellular organelles such as mitochondria. 1.3 Toxicokinetics and toxicodynamics. Two core concepts guide toxicology: toxicokinetics and toxicodynamics. Toxicokinetics describes what the body does to a chemical: absorption, distribution, metabolism, and excretion. For example, inhaled pollutants may enter the bloodstream, be metabolized in the liver, and excreted through urine. Toxicodynamics describes what the chemical does to the body, the interaction of toxicants with biological targets, and the resulting adverse effects. Understanding both processes allows us to predict dose-response relationships, which remain the foundation of toxicology. 1.4 General toxicology and risk assessment. General toxicology examines how different chemicals exert harmful effects, ranging from acute poisoning to chronic diseases. Risk assessment is a systematic approach that asks four main questions. Hazard identification: what health problems are caused by a substance? Dose-response assessment: at what exposure levels do effects occur? Exposure assessment: how much are people or ecosystems exposed to? Risk characterization: what is the overall likelihood of harm? This framework is essential for regulation and policymaking. 1.5 Exposure assessment and biomonitoring. Exposure assessment seeks to quantify how individuals or populations come into contact with toxicants. Sources include air, drinking water, soil, food, consumer products, and occupational environments. Biomonitoring involves measuring toxic substances or their metabolites directly in human tissues or fluids such as blood, urine, hair, or breast milk. Biomonitoring is especially powerful because it reflects actual internal exposure rather than just environmental concentrations. 1.6 Reproductive and developmental toxicology. Certain toxicants target the reproductive system or developing embryos and fetuses. Effects include reduced fertility in males or females, embryo toxicity or miscarriage, birth defects and developmental abnormalities, long-term neurodevelopmental impairment. Examples include lead, which impairs cognitive development, and endocrine-disrupting chemicals such as bisphenol A, which interfere with hormone signaling. In this module, we have traced the foundations of environmental toxicology, its history, ethical scope, cellular and molecular mechanisms, toxicokinetics and dynamics, risk assessment, exposure evaluation, and reproductive impacts. These concepts form the scientific and methodological base for understanding how toxicants affect individuals and populations.